Hello my dear students, welcome back. In this video, I will be discussing about cloning vectors. In my previous video, discussed about the separation and purification of DNA fragments, how they can be isolated. Now, these isolated DNA fragments, they must be introduced into a vector, and from the vector, they should be introduced into a host. The vector in which the DNA fragments are introduced, that is called as a recombinant vector, and this recombinant vector should be introduced into a host cell for gene cloning. So now the commonly used vectors, most commonly used vectors are plasmids, bacteriophages and cosmids. Three types of vectors. So we will discuss now what are the three types of vectors which can be used. So plasmid vector. Plasmids are those which are extracircular DNA present in any bacterial cell. What is the advantage of plasmid is it carries the antibiotic resistant gene. The advantages of plasmids are antibiotic resistant genes are present. Antibiotic resistant genes are present. That is one advantage which gives resistance to the bacterial cell. And another advantage of plasmid is because of its small size. It can easily enter into the host because of its small size. It has only very few genes, and because of the small size, it can easily enter into the host. And it also has a gene called origin of replication. Origin it is called. It means origin of replication. This is the structure of a plasmid. Origin of replication. This origin of replication is very important for attachment of any foreign DNA. And it also decides the copy number of DNA. Copy number of DNA is how many times the DNA can multiply, how many copies it can make within the host cell. That is called as a copy number. So, if a plasmid carries the origin with more copy number, that will be preferred. Normally, plasmids have single copy number. That means they may produce one or two only. Some will have multiple copy numbers. That means they can even produce about 15 to 100 copies per cell. So it has a high copy number. Some plasmids can even produce up to 1000 copies per cell. 1000 copies they can also produce. This type of is called as a relaxed replication control. This is also a gene, relaxed replication control. This relaxed replication control it has the highest copy number. So it is very important that a plasmid with an origin of replication should have the highest copy number. That will be preferred. So these are all the advantages of having a plasmid as a cloning vector. And its insert size, it is called as an insert size. That means how much a plasmid can accommodate. Generally, plasmid can accommodate about 0.5 to 8 kilobases of DNA. Any foreign DNA, it can accommodate about 0.5 to 8 kilobases. This is called as insert size of that plasmid. That means it can accommodate this length of any foreign DNA. Now, the plasmids which are available naturally are mostly, they may not be useful. So, all the plasmids which are available now for the experiment which are being used, these are called as restructured plasmids. They are called as restructured plasmid or modified plasmids. One such modified restructured plasmid is called as PBR322 after the two scientists Bolivar and Rodrigues. After these two scientists, it is named as PBR322. So there are other also available like you have PBr 325, 327, 325, 327, 328, depending upon when they are discovered or when they are restructured. So all the natural plasmids are restructured plasmids. So this was the first. Similarly, other plasmids like PUC, PUC 19, PUC 101, 101 also. You see after University of California, it is called as PUC 19, PUC 101. Then there is a PSC 101 also. PSC stands after Stanley Cohen, who developed the first cloning vector and first recombinant DNA was constructed. It is called PSC 101. So all these are examples of restructured plasmids. Now let us go to some details about what is the structure of PBr 322. So here we find in PBr 322 there are 4 genes present. One is called ampicillin resistant gene. The other one is tetracycline resistant gene is present. The other one is origin of replication, and ROP is replication of proteins of plasmid. Replication of proteins of plasmid. That is called as a ROP. Codes for proteins involved in replication of plasmid. Now we find the sites of restriction enzymes. Sites of restriction enzymes. How many restriction enzymes are given here? There are 8 restriction enzymes. If we go through one by one, so in a clockwise manner, if we go through, first one is EcoR1, and we know all these restriction enzymes, they are isolated only from bacterial cells. They are produced only in bacterial cells. Few fungi also it is observed, and like yeast also it is observed. So EcoR1 is restriction enzyme isolated from the bacterium E. coli. EcoR1 from E. coli. Then next one we have is CLA1. CLA1. CLA1 stands for second one we have is CLA1 stands for from the bacterium Caryophynon latum. Then hint stands for from the bacterium Haemophilus influenzae. Haemophilus influenzae. Then next we have BAM-H1, BAM-H1. BAM-H1 is from the bacterium Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. That is BAM-H1. Then SAL1. Streptomyces albus. SAL1, Streptomyces albus. Then PVU stands for Proteus vulgaris. PVU from the bacterium Proteus vulgaris. PST stands for from the bacterium Providentia stewarti. From this bacterium Providentia stewarti, this particular enzyme is isolated. So all these enzymes are isolated from their respective bacterium, and we can find their sites. BAM-H1 and SAL1, their cloning site or recognition site is, we discussed, it is a palindromic sequence. So BAM H1 and SAL1, the recognition sequence is located in tetracycline resistant gene. Similarly, PVU1 at the ROP, PST1 and PVU1 at ampicillin resistant gene. And the other three, EcoR1, CLA1, HIN3, all these are not between the two antibiotic resistant gene. So this is what is the structure of PBR 322. Now let us go to the next cloning vector, that is bacteriophage. Bacteriophages mainly here lambda bacteriophages are used because they do not harm the bacterial cell, and bacteriophages will have high copy number and they can clone large DNA fragments. Bacteriophage lambda will have a DNA. It is a linear DNA. You know, in a bacteriophage, head of the bacteriophage, there will be a DNA present in concentric circle. So it is a linear DNA. So large DNA fragment can be attached to this. So the insert size, if we take insert size for a bacteriophage, will be 9 to 23 kilobases. 9 to 23 kilobases. This will be the insert size. This will be the insert size for the bacteriophage. That means our DNA of length up to 23 kilobases can be inserted in the bacteriophage DNA. And then this bacteriophage is infected. It infects the bacterial cell. When it is made to infect bacterial cell, it will inject this DNA into the other host cell. Thus the foreign DNA can be transferred. So advantage of bacteriophages, large DNA fragments can be cloned. Now the third type of vector is called as a cosmid vector. The third vector is called as cosmid vectors, cosmids. Cosmids are also artificially synthesized vectors. These cosmids are prepared from bacteriophage and plasmid. They are the combination of bacteriophage plus plasmid. So here lambda bacteriophages, the DNA from lambda bacteriophages are taken and they are attached to a plasmid DNA. So these lambda bacteriophages are completely harmless. They do not harm the host cell. And here also the large DNA can be cloned. Compared to the bacteriophage, cosmids large DNA can be cloned. So here, if this is a bacteriophage DNA, this particular part, which is called as a cos site. Cos means cohesive site. Cos is cohesive site. This is also a cos site. Towards the end there will be cos sites. So these cos sites only can be taken. So these cos sites can easily join and they form a circular DNA. The remaining part, which is actually not required, this can be removed. And in this place, it is joined with some advantages, some features of plasmid like antibiotic resistant gene and origin of replication. So these two are combined. And now this cosmid will have some features of plasmid and also some of the bacteriophage. Now to this, if this part is removed, now to this, in between, any other new DNA, eukaryotic DNA or any other new DNA can be added here. This is the eukaryotic DNA. Eukaryotic DNA or any other foreign DNA. And this part is consisting of, this is the cos site on both the ends, which is taken from the bacteriophage, and remaining are the features of the plasmid. These cos sites can easily join and they can form a circular DNA. Now, this such restructured DNA, this is any other foreign DNA. Such restructured DNA will be now introduced into the phage capsid. It will be now introduced into the phage capsid. You know the phage capsid will have the head part and the tail part. So into that this circular DNA will have some part of plasmid and it will also have the eukaryotic DNA or any other foreign DNA along with this. Now this will be infecting the bacterial cell. This will be infecting the bacterial cell. Bacterial cell will have its normal DNA. So when this infects the bacterial cell, it is a lambda bacteriophage. Since it is a lambda bacteriophage, it is not going to cause any harm to the bacterial cell. So this will directly introduce its DNA into the cell. So DNA will be injected into the cell and here we find the foreign DNA enters into the bacterial cell. Now this foreign DNA will be able to multiply within the host cell. So the advantage of using plasmid is much larger DNA fragment can be cloned and insert size for plasmid is about 30 to 45 kilobases. That means maximum up to 45 kilobases, the DNA can be inserted. That is the advantage of a plasmid. So cloning of large DNA fragments can be done by using plasmid. So this is useful in constructing many genomic libraries like human genome library that is constructed by using this plasmid. So large number of fragments can be cloned. That is about what is the different types of cloning vectors. So here I conclude my video, my dear students and other viewers. So in my next video, I'll be starting with, we have after cloning vectors, we have the next topic as what are the different features that are required for a cloning vector. So this I am going to complete it just in this video and then we can conclude this video. Because now that we have done what is a cloning vector, now let us see what are all the features which should be present in a cloning vector. So first feature it should be present, any vector which can be used. So next topic what I am going to discuss is the features of a cloning vector. Features of a cloning vector. So first feature of a cloning vector is presence of origin of replication. Origin of replication is very important for a cloning vector because already as we discussed that decides the copy number. So a plasmid will have the origin of replication with highest copy number. That's why even out of these three, plasmids are generally preferred because they have origin of replication and that can be altered. So origin of replication is useful in initiating attachment of foreign DNA and also decides the copy number. Now next important feature is presence of, second one is presence of a selectable markers. Presence of a selectable marker. The meaning of selectable marker is that marker should be useful for selection of recombinant cells from non-recombinant cells. What is the meaning of recombinant cell? A recombinant cell is one which is carrying the foreign DNA which has to be cloned. That is called as a recombinant DNA. Non-recombinant will not carry that gene which has to be cloned, or DNA which has to be cloned. So again here plasmids will have antibiotic resistant genes. They have antibiotic resistant genes. So these antibiotic resistant genes will function as selectable markers. Just like we have seen in PBR 322, depending upon, there are some genes located on located on the tetracycline resistant gene, and some genes are located on ampicillin resistant gene. Suppose we use those enzymes, those cloning sites of the enzymes like BAM-H1, it is located on tetracycline resistant gene. So if we use the enzyme BAM-H1, that means the tetracycline resistant gene is getting inactivated, and the cells will not grow in tetracycline medium. They can grow only in ampicillin. So that's how they can be separated from recombinant and non-recombinant, from transformants and also from non-transformants. That we are going to do in the other topics where selection of these cells is done. So plasmids will have antibiotic resistant gene. What is the function of antibiotic resistant gene? They help in separation of recombinants and non-recombinants. Separation of recombinants and non-recombinants. Also transformants, that means which have undergone transformation, and non-transformants, and non-transformants. So that is the function of these selectable markers. Okay, next we have is the next feature of a cloning vector is presence of cloning sites. Third one is cloning site. What is a cloning site? Third one is a cloning site. A cloning site is a recognition site for any particular restriction enzyme. Like we have seen in the PBR 322, there are about 8 cloning sites shown. And each cloning site only one is present, not more than one. So that is one feature of a cloning site. So cloning site, only one cloning site for a restriction enzyme must be present. One cloning site for a restriction enzyme must be present. If more than one is present, say for restriction enzyme, say suppose EcoR1 we take. If we have more than one, then there will be confusion in the number of fragments obtained. Many similar fragments are obtained. Like EcoR1 will recognize the cloning site GAATTC. This is the cloning site for EcoR1. So when EcoR1 has more than one cloning site, similar fragments are obtained, and it will be very difficult to identify a particular gene because DNA is a very long molecule, and we have to identify a particular gene and then we have to clone that gene. First we have to select that gene and then we have to clone it. So this step by step we are going to discuss in the next topic, which we have is the what is the technique of rDNA technology. So again we will be listing down different steps there. So there should be only one cloning site to avoid any confusion regarding selection of fragments. That is what is the feature of a cloning site. Again, with respect to cloning site, PBR 322 or plasmids are considered to be the best cloning vectors. Though the other vectors are used only because their insert size is more. Otherwise, plasmid has many advantages compared to other cloning vectors. So with this, my dear viewers, we complete different types of cloning vectors. So next we have to, I think there is one more, let me check. There is one more cloning vectors which is present. Let us complete these two also, cloning vectors. So till now I discussed about bacteriophages and then plasmids and cosmids and different features of cloning vectors. There are cloning vectors separately present for plants and animals. Introducing into plants and animals, we have different cloning vectors. So cloning vector, let us see for plants. Cloning vectors, let us see for plants. In plants, the most commonly used cloning vector is Agrobacterium. Agrobacterium. This infects all the flowering plants, causing cancer-like growth, tumor-like growth, causing the disease. Agrobacterium tumefaciens. Its species name tumefaciens, and it causes the disease called crown gall disease. It causes the disease called crown gall disease. And because the plasmid which is present here is called as a TI plasmid. TI stands for tumor inducing plasmid. It is called TI plasmid. This TI plasmid has a particular DNA which is called as, this DNA which is present, it is called as a tDNA. The DNA is called as a tDNA, tumor inducing DNA. This part of the plasmid actually causes the disease. So, if it is used as a vector, cloning vector, what is done is, this tDNA is removed from this TI plasmid so that it will not cause any disease. And in its place, a new DNA can be inserted. So, here tDNA is removed and in its place, any other desirable DNA can be introduced. Any other desirable DNA can be introduced. And this can be made to directly infect any dicot plants. So that we are directly introducing this new DNA into the plant. So through this vector, we can introduce the new DNA, foreign DNA directly into plants. And this type of pathogen, it is a pathogen. But what are we doing initially? We are removing the disease causing gene. So these are called as such pathogens. These are called as disarmed pathogen. It is the disarmed pathogen. So its virulence is taken away, virulence is lost. Then it can be used as a vector. That is in case of plants. Now, in case of animals, retroviruses are used. Retroviruses will infect the plants, will infect the animals. Retroviruses are RNA viruses. So, same thing it is done. From the retroviruses, the disease causing part of the RNA, disease causing fragment of the RNA, that is removed. And then that can be used. In that place, whatever foreign DNA, whichever DNA or gene has to be introduced, that is introduced. Then it is made to infect any animals. So directly this can be, foreign DNA can be directly transferred to the animal. So retroviruses for animals and agrobacterium for plants, directly they can be used as a vector without any bacterial cell. Here we are not using any other host bacterial cell. Directly we are taking the plasmid DNA, and this plasmid DNA from agrobacterium, suppose this is a modified plasmid. So this plasmid, first it should be reintroduced into agrobacterium. Then this agrobacterium will infect the plants. Of course, plasmid cannot directly enter into any eukaryotic cell. So it should first enter into the agrobacterium and then it should be made to infect this plant. Similarly, retroviruses will directly infect the animal cells. So with this, my dear viewers, I think we almost completed up to what are the vectors. So I conclude my video here. In my next session, I will be starting with the other topics like competent host, how are the host used, and then we have about say technique, different process of rDNA technology. So thank you very much all my dear students and all my other subscribers, all my viewers. So I want you all others to share these videos and the link to the maximum, so that I want to reach the maximum people, maximum students who can get benefited by this. So please subscribe my channel. After watching, then share and even ask all your contacts. Share the link and ask them to subscribe. Thank you very much. Have a good day.